Hey guys, welcome to week 3. We're going to go over cells and their function today. We're going to get through as much as we can. If we don't get through it all, we'll cover what we didn't do next week. But I'm going to try to fly just like last time. So we just did chemicals. We did our atoms and our molecules. Now we're going to jump up to cells. Cells are made up of the chemicals that we were talking about earlier. Also, the chemicals we were talking about earlier pass through the cells, and we're going to talk about how that happens. So cells, you can see cells if you have a microscope. You can see them, and then cells are alive, okay? So this is our smallest living unit of matter. It's made up of 3 different things. You have the membrane, the cytoplasm, and the nucleus. So first we're gonna go over the plasma membrane. I can share my screen. Okay, so the plasma membrane is made up of lipids. It's called the phospholipid bilayer. One side of this layer is, each side of these layers facing the water have hydrophilic heads of phospholipids pointing towards the water. So you can see water would be in here, water would be on the outside, and these hydrophilic heads face the water, while inside you have your hydrophobic tails. So hydrophilic heads face water, hydrophobic tails face inside where they can kind of huddle together and make a barrier so no water can get inside. So this is obviously very exaggerated. It's very enlarged to show that detail. But this is our first thing. This is our plasma membrane. I want you to highlight the functions of the plasma membrane, the physical barrier to prevent things from coming in. So if, let's say, we had a water-soluble molecule coming and it wanted to get through into the cell, this water-soluble molecule would hit that hydrophobic tail and bounce out again. Okay, it makes a barrier so water-soluble things can't get through because it has that hydrophobic tail, and then lipid-soluble things cannot get through because it has the hydrophilic head. Okay, so it acts as a barrier. It controls the entries and exits like we just went over. It has a bunch of proteins that we're going to go over that helps it respond to its outside environment. And then it provides structure. So we'll also go over, that's another protein thing we'll go over. But mostly, I'd want you to know those functions for sure, but then go over the phospholipid bilayer. Know which side is hydrophilic, which side is hydrophobic, and then why that structure is that way. So it prevents the passageway of things, and then it also creates this barrier. Okay, inside here, it doesn't have a picture, but if I were to draw right here, there's just little cholesterols, little pieces of cholesterol that are stuck in this hydrophobic area, and that creates a little bit of rigidity to this phospholipid bilayer. That way it doesn't just like move and flow, it kind of keeps its shape. So this cell would be a circle. It's because those cholesterols in that hydrophobic tail would be holding things in place. Then we have proteins. I'm going to get to my proteins. So this is not all of the proteins, it's just the best picture I could find. So I'm going to draw. Okay. So proteins, these you can see here, you have your phospholipid bilayer. These proteins are embedded inside that bilayer. So a few of these proteins, I'm going to just start via your manual. So your anchoring proteins. I like to think of these as your Velcros. Okay, so if it wants to anchor itself to a wall or another cell or a bacteria or something, these little Velcro proteins or anchor proteins will grab on, and then it will be anchored to whatever is right here. So this one, I don't know what it's anchoring onto in this picture, but it anchors the cell to something else. Recognition proteins, here they're called receptor proteins. Sorry, we actually don't have a recognition protein. So I'm going to draw myself a little recognition protein. What this does is it has on it a little protein end that if something were to come, another cell were to come with a protein, that it had the same shape as that recognition proteins, it can determine what this cell, so let's say this is another cell over here. Oh, it's not letting me draw, that's okay. So this recognition protein is going to help determine if this cell over here is self or foreign by recognizing a protein or an antigen on its plasma membrane. So let's say this is a bacteria that's coming up to this cell. This recognition protein would be able to bind on to that antigen and say, this is a bacteria, this is not good, and it will signal a response, okay? So recognition proteins recognize that it's either another self cell or it's a foreign cell, like bacteria. Enzymes is our next protein. They just cause reactions to occur. So you learned about enzymes last chapter. Hopefully that's a review. We just find them embedded in plasma membranes sometimes. Receptor proteins, they will take things floating around in the fluid on the outside, and then they will receive them, or it's kind of like a lock and key, that thing that's floating around. It could be a chemical, it could be an antigen. It will come, lock into this receptor protein, and that receptor protein will be like, oh, I know what this is, and a reaction will occur inside the cell. So this is my, like to think of a lock and key. So you put the key in, it causes something to happen on the other side of the cell. Carrier proteins, here they're called transporter proteins. They just carry things either inside the cell to outside the cell or vice versa. So they're just going to be transport, because remember how we talked about that phospholipid bilayer, nothing was able to pass through. So these carrier proteins or transporter proteins just allow a passage. They transport things either inside the cell to out or outside to in. Channels is just kind of like it says, instead of having the protein do the work, it just creates this hole where the solute can just pass through freely. Okay, so channels don't require any energy or anything. They're just a hole to pass through if needed. Okay, so anchoring proteins is Velcro. Recognition proteins recognizes self or foreign cells. Enzymes catalyze reactions. Receptor proteins grab on, it's your lock and key, receive something and it causes a reaction to occur inside. Carrier proteins, they transport things inside the cell to out or outside to in. And channels are just holes in the cell so things can pass through. You also, on your membrane, you have carbohydrates and they come out, they're like little sugars. Well, it's not letting me draw, that's okay. So little sugars that kind of stick out and they have them embedded all over in the membrane. This is called your glycocalyx film. So this just causes like a sticky surface that can grab onto things and pull them into your proteins for the proteins to use them. Okay, so those are your proteins. Know the functions of your proteins there. Okay, the next structure of your cell, let's see. Oh. Going in. Okay, we are going to start going over all of the things that are found actually inside the cell. So we just went over this membrane. You can kind of see it's that double layer, phospholipid bilayer. Okay, all of your organelles or the functional units inside the cell are suspended in what we call cytoplasm. This is literally just like Jello. It's like bananas and Jello. So all of these are the bananas just floating inside the Jello. That cytoplasm just holds things in place. Okay, the organelles are found inside this Jello or this cytoplasm. Okay, your cytoskeleton, that's shown by these like microtubule blue things. You don't need to know microtubule. It just holds the inside of the cell. So remember, you have the cholesterol holding the phospholipid bilayer in that structured shape. Inside the cell, just making sure that it doesn't collapse in on itself, you have your cytoskeleton. So your cytoskeleton just maintains kind of that shape of the cell. Back up just a second. New share. There we go. Okay, so on your cell, so here you have your phospholipid bilayer. I don't want to keep drawing it because that's going to take forever if I make this super long. So I'm going to just draw your phospholipid bilayer as a line. Hopefully you guys understand what that means. So this is just, I didn't want to keep drawing it. But here's your membrane. Sometimes this membrane is flat, but sometimes your membrane gets these folds in it like this. I didn't mean to do those gaps. So sometimes your membrane, these phospholipids line up nice in a straight line, and sometimes they get this wave motion going here. This is called your microvilli. And what this does is it increases the surface area of the outside of your cell. So if I had this much of a membrane between this point and this point, and it's flat, that's how much surface area I have for absorption. But if I make it super bendy from here to here, same distance, look how much more membrane I have between like point A and point B. I have just more surface area for absorption. So this is your microvilli. Microvilli. Oh, whoops. I don't know what's happening. That's okay. I won't write microvilli because I'm struggling, but that's okay. So microvilli increases the absorption of things by increasing the surface area. So more solute, so I have this solute, it has more places on this membrane it's able to hit to be able to be brought into that cell. Okay, this is found a lot in your intestines because you eat your food and it breaks down and you want to absorb all your nutrients, and that's done through these little microvilli. Okay, so hopefully that made sense. Go back to my cell here. Okay, centrioles are our next thing. I think of these as like two little pieces of firewood. Okay, so centrioles are in charge of cell division. What these centrioles do is they shoot out. You would have a centriole on the other side over here as well that's covered. So you'd have two, one at this end, one at this end. And what the centrioles do is they shoot out what we call spindle fibers. That's an important word to know, spindle fibers. So it shoots out the spindle fibers and they grab on to all of the organelles and all of your DNA inside your nucleus. These spindle fibers grab on and then they're going to yank. So they grab on, they hold on to all your organelles and that DNA in the middle, and then they're going to yank. You have some spindle fibers coming from one side, some coming from the other side. They grab on and then they pull. Okay, and as they pull back, I'm going to draw it right here. So as they pull back, you can imagine you're going to have a division in your cell. So they pull back so hard that it actually splits that cell in half. So then you're going to have two new cells. Okay, so your centrioles are in charge of cell division by shooting out spindle fibers that grab onto all of your cell contents, and then it's going to yank it splits them evenly, yanks it from side to side until that cell divides into two new cells. Okay, so if you don't have if you don't have centrioles, you won't be able to perform cell division. Okay, centrioles use those spindle fibers. Spindle fibers are what actually grab on, hold, and pull. That can be kind of confusing. Email me if you have any questions about that. Erase this. Okay, moving on. We have our cilia. So just like we have microvilli, we also have things on our membrane called cilia sometimes. These two, microvilli and cilia, get mixed up all the time, so don't get them mixed up. I'm warning you right now. Okay, cilia are hair-like projections on the outside of a membrane. Oh, it's not letting me draw. There we go. They're hairy, it's like a hairy cell. Okay, so it's not folds in the membrane like our microvilli was. It's hairs on the membrane of your cell. Okay, that's not the actual membrane folding, it's actual physical projections coming out that are hair-like. And what happens is these little hairs, or these cilia, they move together. They're like at a concert, if everyone was like doing the wave, that's exactly what they do. Okay, so they stick up their little arms and they do the wave. They push on, so they'll grab onto something and they'll wave and they'll push it to the cilia that's next to them. And that cilia will push it, and they all beat together to move things. So it's like if someone was body surfing, these little cilia would push them along, and that whatever the particles moving would be able to move along that cilia as that cilia beats for that movement. These are found in your lungs. So think of all the mucus and junk that would get stuck in your lungs. Your cilia get that mucus and beat it up towards your throat so you can swallow it. This is why when you smoke, if you can burn the hairs, they say hairs, it's your cilia that's burning in your lungs, and you're not able to beat that mucus out of your airways, and you get a lot of infections. Okay, so that's cilia. Don't get microvilli and cilia mixed up. Microvilli is for absorption, and it's the physical membrane folding, and the cilia is hair-like projections that beat to move something across a membrane. Okay, good on that. Next, we're going to go to ribosomes. Ribosomes make proteins. They are where all of the protein production happens. Ribosomes on this picture are portrayed as these little red dots that you see all over the place. Proteins are, as we talked about last time, how our body functions. We need proteins for most everything we do. Okay, and these ribosomes are responsible for creating those proteins. They are the site of protein production. It's called production. Another word for production is synthesis. So you'll hear it called protein synthesis. That happens inside these little ribosomes. Some of them are free floating, like these little ribosomes shown here, and some of them are fixed on our rough endoplasmic reticulum. The endoplasmic reticulum is actually called rough because these ribosomes are stuck to it. It gives it a bumpy appearance. Okay, so ribosomes make proteins. You have free ribosomes floating right outside that nucleus, and fixed ribosomes stuck on the rough endoplasmic reticulum. Moving on to your endoplasmic reticulum, you have two. You have rough endoplasmic reticulum and smooth endoplasmic reticulum. The difference literally is just that one has ribosomes on it and one does not. Okay, so rough endoplasmic reticulum has ribosomes, smooth endoplasmic reticulum does not. And that completely changes their function because of those ribosomes. So we're going to start with smooth ER. You'll hear it called ER, smooth ER, endoplasmic reticulum. This is the site of production of pretty much everything else except for proteins. Because remember, ribosomes make proteins and smooth does not have ribosomes. So there is going to be no protein production in the smooth endoplasmic reticulum. Okay, but you're going to make fats, you're going to make steroids, you're going to make glycogen, all of that happens in your smooth endoplasmic reticulum. Okay, so if you get a question that asks if smooth endoplasmic reticulum makes proteins, the answer is no, because it does not have ribosomes, the site of protein synthesis. Okay, but it makes almost everything else. Let's move on to rough ER. So this rough ER is found closer to the nucleus, and it is the site of protein synthesis. So Your DNA here, we'll talk about this later, releases information into these ribosomes to make proteins. Okay, so the ribosomes make the proteins, and then the rough ER is going to fold those proteins. So if you remember from last chapter, you have your primary, secondary, tertiary, quaternary structures. We didn't really go over secondary, but that folding, so it started as a chain of amino acids, then it folded into either spirals or folds, and then those combined to either make a globular protein, that's just a bunch of swirls or folds mixed together, or fibrous proteins, which is a bunch of folds. Okay, hopefully you know. That folding, this process of going from a chain of amino acids into a functional protein, occurs in the ER. So ribosomes make the protein. It then goes into the rough ER where it's folded into its secondary and tertiary structures. And then it's going to get released into your Golgi body. Okay, also known as the Golgi apparatus. Okay, so that's super important. Your rough ER folds proteins and sends them off to the Golgi apparatus. I like to think of the rough ER as kind of the packaging company. It's FedEx. It takes the product from the ribosomes, folds it nicely into a box, and sends it off to the Golgi body. Okay, so rough ER, the chain of events, it just folds and sends it to that Golgi body. Moving on to our Golgi body. Our Golgi body is probably has one of the most complicated jobs in our cell. So our Golgi body or apparatus, you can say it either way, reminds me of like stacked dinner plates. Okay, they just stack up nicely. And then we just talked about the rough endoplasmic reticulum sends little packages to the Golgi body to be transported off. So I'm going to open a new share real fast. So you can draw your Golgi body here real quick. There we go. Okay, so here's your Golgi body. Your rough endoplasmic reticulum just sent these folded up proteins. So here is a protein that has been sent in. Okay, so this protein's going to come into your Golgi body where it's going to be packaged into a vesicle. So it passes through the membranes of the Golgi apparatus, and it will come out on the other side with a nice little package wrapped around it. Okay, so it packages those proteins that were sent from the rough ER. Okay, that's why it's called one traffic director. That's just what it's doing. It's taking these proteins, putting them in a nice bubble, and telling them where to go. Okay, that's also number 2. It packages these proteins and sends them wherever they need to go. So let's say this is your outside of your cell. There. Oh no. It's super annoying. There we go. Okay, so this is the inside of your cell. This is the outside of your cell. One other thing this Golgi body can do is it can actually repair this membrane. So let's say we got a hole in our membrane. Oops, it erased the whole thing. That's okay. We have this hole in our membrane right here. It can, just like it made this little vesicle to hold the protein from the rough ER, it can make a little vesicle that's actually just empty. Okay, so it just makes this vesicle that's empty. That vesicle is then going to transport over to the plasma membrane, and it's going to morph itself into the membrane and fix itself. It makes a patch. Okay, so the Golgi body secretes these vesicles that are empty to go and repair the plasma membrane. Okay. One other thing that it does is it can create what we call a lysosome. So just like you had an empty package, a package with a protein, you can also have a package with a bunch of like poison inside of it. So here we go. This package, it creates this lysosome juices. I don't know what you'd call that. Cytosol. The lysosome, this poison inside, the Golgi creates that and then packages it off, okay? And we're gonna go over what lysosomes do more. But those are the three things that happen in the Golgi. You get proteins from the rough ER, they're packaged and either sent outside of the cell or somewhere else inside the cell. It can also send off empty vesicles to go repair your membrane. And then it creates this poison, wraps it in a little vesicle, then sends it off, and that creates a lysosome. Now we're going to go over what lysosomes do. Lysosomes are how we destroy broken things or foreign things. Okay, so they're in charge. They're like our little garbage man. So I'm going to make another little lysosome here. So here's our lysosome. It can do one of three things. So let's say we have a broken organelle. We're going to have a broken ribosome. Our ribosome is broken in half. Here's a ribosome. This lysosome can come up to your broken ribosome. It will fuse with it and then break down that broken ribosome until it's just left with its parts, and then those parts can be recycled. Okay, so that's the garbage man function. The ribosome broke, the lysosome came up, fused with it, and got rid of the broken ribosome. Another thing that the lysosome can take care of is bacteria. So let's say we've got an invader in here, bad guy. This lysosome, just like it did with that broken ribosome, can fuse with the bacteria, release its poisons onto the bacteria, and kill the bacteria. Okay, so once the poison gets on the bacteria will die, and then the cell can reuse its part, recycle its parts. Okay, and then the third thing that the lysosome can do is the lysosome can actually pop itself and release a bunch of that poisonous fluid into the cell. If that happens, the entire cell will die. This is called, this word is super important, autolysis. Auto means self, lysis means destruction. So the lysosome pops and the poisons go all over inside the cell and kill the entire cell. This is really great. Let's say you have a cancer cell that has started mutating. If your lysosome pops, it can actually kill that cell before it can mutate into other cells. So autolysis is really important for controlling the growth of abnormal cells. Okay, so three things. It can either destroy broken organelles, it can destroy bacteria, or it can go through autolysis and kill the entire cell if the cell has begun malfunctioning. Okay, let's go to mitochondria. Mitochondria. So back to our cell. Mitochondria, I'm sure if you remember anything from high school, you'd know that mitochondria is the powerhouse of the cell. You probably don't know what that means, you just know that it's the powerhouse of the cell. So our mitochondria look like little beans with a little maze inside. They create energy. Energy is ATP. Okay, so those can be used interchangeably now. ATP and energy are the exact same thing. Okay, ATP is energy. Mitochondria create this energy or this ATP. Coming back to my whiteboard here, we're going to go through what actually happens inside your mitochondria. Okay, so here I'm going to draw myself. Mitochondria. Okay, here's the little mitochondria. If you remember back to the last chapter, we have glucose. Glucose is how we make our energy. Okay, so mitochondria, you have one glucose come up to a little mitochondria. Before entering the mitochondria, that's super important, outside, right outside the mitochondria's door, your glucose is going to break down into two pyruvate molecules. Okay, so glucose breaks into two pyruvate molecules. Okay, this process is called glycolysis. That's a word that you're going to see a lot. So this is called glycolysis. Glucose breaking into two pyruvate molecules outside of your mitochondria is called glycolysis. This does not take any energy. This does not take any oxygen either. Okay, so this is what we call anaerobic, meaning that it does not require oxygen. No oxygen is required for glucose to break into two pyruvates. Okay, then your pyruvate, I'm only going to draw one for the sake of simplicity going in, but both pyruvates are going to enter into your mitochondria. Okay, so the pyruvate enters and goes through what we call the Krebs cycle, or also known as the citric acid cycle. So this is called your Krebs or citric acid cycle. Okay, so this pyruvate goes through like 20 something different chemical changes. You don't need to know those. You just need to know that it goes through a cycle where CO2 is released. So out of this Krebs cycle, you're going to have CO2 released. Oxygen comes in. Hydrogen is released. And you have lots and lots of ATP is released. Oops. So you don't need to know the number, but there's like 30 something ATP released from this Krebs or citric acid cycle. Up here in glycolysis, I should have mentioned this earlier, you have 2 ATP that are released. Okay, so glycolysis does produce energy, but it's very, very small compared to the Krebs or citric acid cycle that occurs in the mitochondria. So again, going through this, one glucose molecule breaks down into 2 pyruvates, releasing 2 energies or ATPs in a process called glycolysis. This does not require any oxygen, so it's called anaerobic. That pyruvate molecule, both of them, but just one, is going to, for the sake of the drawing, is going into the mitochondria, where it goes through a series of reactions called the Krebs or citric acid cycle. Oxygen is required for this to work, so it is called aerobic, meaning oxygen is required. Hydrogen and carbon dioxide are released, while lots of ATP are also released. So there's like 32, I believe, that are released. You don't need to know that number, but a lot of ATP are released from this Krebs or citric acid cycle. This oxygen is actually going to combine with the hydrogens here, and you're going to form H2O, also known as water. Okay, so there's lots of things that happen in this Krebs cycle. You just need to know that oxygen is required. The pyruvate is what goes through the citric acid cycle. Most of your energy is going to be made here. And then you have a byproduct of carbon dioxide and water. Okay, so one pyruvate makes a lot of ATP, water, and carbon dioxide. Watch videos on this. If you get to things about the electron transport chain, that's just more steps. Don't listen to that. You don't need to know that. This is just simplified into what you do need to know. Okay, let's go to the nucleus really fast. So your nucleus, this is the brain of your cell. Okay, inside your nucleus, you're going to find DNA. DNA or genes are what make up who we are. So you have the same DNA in every single cell in your body. Some of that DNA is just turned on, and while others is turned off. That's why different cells perform different functions. It just depends on what part of the DNA is turned on and what part is turned off. Okay, and this is found in your nucleus. So inside, here's your nucleus. Inside, you can either have, here's another nucleus, DNA that is swirly like a plate of spaghetti. So your DNA can either look like this, or your DNA can arrange nicely into little X's. Okay, this spaghetti-like DNA is called chromatin. Chromatin. This is found when the cell is not going to divide. Okay, when it's arranged nicely into an X, it is called a chromosome. Chromosome. Chromosomes are found in a nucleus that is going to be divided. So we talked about those spindle fibers from your centrioles that grab on and pull the cell apart. So those spindle fibers are actually going to grab on to each side of the X here and pull it apart so you can split that X right down the middle. If you tried to do that with your chromatin, this plate of spaghetti, do you think you would split it directly, perfectly in half? It would be very, very difficult. So that's why the chromatin does not divide. When a cell is about to divide, this chromatin coils really tight and actually becomes a chromosome. Okay, so chromatin does not divide while chromosomes do. They're the same thing. One is just coiled and one is not. One is like a plate of spaghetti. Okay, DNA is your genetic code. So you have three things. You have your genome, you have a gene, and you have your genetic code. Your genome is every single piece of DNA in your body. Every single letter in your book is kind of how I like to think of it. So I like to think of a genome as a library. It makes up your entire person. You have 46 chromosomes with millions of little letters of DNA in there. Okay, this is who you are. You are your genome. It's what makes you you. Okay, your gene is going to be your one protein worth of information. So I like to think of this as a sentence. Your sentence makes one protein. So this is just a string of about 100 letters that we talked about before, like your A, T, C, Gs that we talked about last class. This is your genes. It's those letters put together to form a sentence. That sentence is going to create one protein. Okay, so your gene, let's say your sentence is, she has blue eyes. Your protein is then going to be formed to make you have blue eyes. So it's just one sentence in the library that makes you you. Okay, so you've got thousands and thousands of genes. So I have a gene that says I have brown hair. My sentence says you have brown hair. My gene says you are very white. That would be one sentence. Okay, so your gene is just one specific sentence or one specific code for a protein. Your genetic code is going to be the sequence of those letters. So it's the process of putting together your A's with your T's and your C's with your G's in a very specific order. So these are the letters in your sentence. Okay, so gene is a sentence, genetic code is that letters that make up the sentence. Okay, hopefully that makes sense. Library, sentence, letters, it kind of helps me think about it. Hopefully that helps you too. Okay, now we're going to go into protein synthesis. This is probably the most confusing part of this chapter. Watch lots of videos on it. It helps. So inside your cell, let's say you have a DNA that reads A, T, C, G, T, A. You know, being your smart person, that A pairs with T pairs with A, C pairs with G, G pairs with C, T pairs with A, and A pairs with T. Okay, so here is your string of DNA. When your body decides that it's time to make a protein, this is going to unzip. Okay, so it literally unzips like a zipper, and one side goes away while the other side stays. Okay, so let's say we unzipped and got rid of our bottom half. So we unzipped it and took away this bottom portion. So we're left just with the top portion. You then have your mRNA that's going to come in letter by letter and connect to this exposed side of DNA. Okay, so your mRNA, your messenger RNA, comes in letter by letter and is going to attach to this DNA strand that was unzipped. So you know from last chapter that RNA does not have any T's in it. Instead of T's, it's replaced with U's in RNA. So instead of this A being paired with a T, it's going to be paired with a U. Okay, and then this T is going to get paired with an A, because you can have A's in RNA, just not T's. Then C is going to get paired with an RNA of G. G is going to get paired with the C, T with an A. And then again, we have an A. A is only going to get paired with a U, because T's are not found in RNA. Okay, so this is going to be our oops, oh man. mRNA. Okay, so this is just, they came in letter by letter and attached to that exposed DNA that was unzipped from its other side. Okay, this mRNA just codes letter by letter to that DNA and makes its own chain. This is called transcription. Transcription. You're literally transcribing the RNA from the unzipped DNA. Okay, then what happens is this mRNA is going to float outside of your nucleus onto a ribosome. We talked about how ribosomes make proteins. We're just going into specific detail about how that works. So your mRNA floated outside the nucleus, came over and attached onto a ribosome. Okay, starts at one end first. Okay, so the mRNA attaches onto a ribosome. Label this real quick. Ribosome. Then what happens is you have what we call tRNA that comes into play. So your tRNA is kind of special. It connects, it literally looks like a T. And it has on top of it an amino acid. Okay, so this tRNA is going to be an A tRNA. So A. You know that A pairs with U, so this tRNA is going to come and attach to this U. Okay, so this floats down and attaches to this U. Then you have another tRNA come along with a different amino acid on top that is going to connect with this A. And you know that the A connects to U in RNA. Oops, I didn't mean to make that pink. A connects with U, so that U is then going to attach onto that A. And the same thing's going to happen. This mRNA is going to kind of slide through this ribosome, and every time you get to the center of the ribosome, a tRNA is going to bring an amino acid over to the ribosomes by connecting letter to letter that matches. So A to U, U to A, G to C, C to G, U to A to U. Okay, and then once that tRNA detaches, these proteins, these amino acids are going to connect together. Okay, and as you get more and more tRNA that come in and connect, you're going to get a sequence of amino acids that all connect in a chain. Okay, so once this is complete, you're going to have just a chain. I'm not going to do all the colors for sake of time, of amino acids in a very specific order that was determined by this mRNA. This is called translation. Translation. Okay, so the transcription occurred when the DNA unzipped, the mRNA came and attached on to the exposed DNA, coding what that DNA had said. The mRNA then leaves the nucleus and transports to a ribosome, where that ribosome is able to hold the mRNA so the tRNA can come and attach on top with the amino acids hooked to that tRNA. And the amino acids are then going to bind together in the sequence determined by that tRNA until you have a protein chain. That's translation. Okay, so making that protein, making DNA into mRNA, then having tRNA connect to that mRNA to make a protein chain is called protein synthesis. So this whole thing is called protein synthesis. Oops. This is how our body is made. So our DNA tells our body what to do, and our mRNA takes it outside until we make the proteins that are coded from our DNA. Watch videos on this. This can be super confusing for people. Watch videos. Watch this again if you need. Email me. Whatever you need to clarify this till it makes sense. Okay, one thing to know under translation, it says proteins. You will have a question that kind of goes over, it's kind of a review question. Remember, enzymes are proteins. Channels, like we talked about earlier, are proteins. Antigens are proteins. If you were asked what glucose was, that's a carbohydrate. That is not a protein. So if it asked, does protein synthesis make glucose? You would say no. But these three things are proteins. Okay, let's move on to membrane. We are flying. We're actually going to get this done. Clear this. Okay, your cellular transport. So we have a cell membrane here. Here is our cell membrane. On our cell membrane, we have a few ways to transport things across. You can either have a membrane that's completely solid without any holes in it. That's going to be impermeable. Some of your membranes are going to have just free channels, allowing anything that wants to pass in or out. This is going to be your freely permeable. That just means anything can pass through that wants to pass through. You are then going to have some that have very picky channels. So this is going to be our hydrogen channel. Only hydrogens can pass through a hydrogen channel. If I had a sodium come by, it would get rejected. It could not pass through this hydrogen channel. Membranes with these kind of channels are called selectively permeable. They select the only thing that they're going to allow in. So completely solid is impermeable. Open channels that allow anything in and out is freely permeable. And then very picky channels that only allow certain things in create a selectively permeable membrane. Okay, so now we're going to go over diffusion. So I want you to picture real fast a room. So you're in a room and it has 50 people packed inside there. It's a small room. You are shoulder to shoulder with people. Over in the next room over, you have a room with only 2 people in there. The room with 50 people inside of it, it's getting hot and sweaty and there's too many people and you're bumping into each other. You're going to want to pass through to the room that only has 2 people in it. Just like that, we have molecules want to, if one side is full or has a large number of molecules and the other side only has a few, the molecules that have a large number are going to want to pass to the side that has a low number. Just like you wanted to get out of that hot, sweaty room to the room that only had 2 people, molecules want to do the same thing. They want to move until they get to be an equal number in each room. So 1, 2, 3, 4, 5, 6, 7. So in this case, there would be 5 on each side. And then the movement would stop because you're happy having the same number on both sides. Okay, this is called diffusion. The molecules diffuse across the membrane. So this is diffusion. Osmosis is the movement of fluid. So let's say, same scenario, you're packed on one side. You've got a lot of molecules on one side. I'm going to actually close this off here. Okay, so let's say I'm packed with molecules on one side. And on the other side, I just have a few. But this membrane is impermeable to solutes. It's only going to allow fluid to move. What's going to happen? Think of your hot, sweaty, small room that you're in with your 50 people. Another solution to making this situation of being cramped into one room, better is to make that room bigger. Because if you make the room bigger, then the molecules have more room to spread out. So that's what water does. Water is going to move from the side with fewer to the side with greater. So water is going to be purple in this case. And it's going to move to make whatever side had the more molecules bigger. So now 1, 2, 3, 4, 5, 6, 7, 8. Now, instead of being packed so tightly together, these molecules are as evenly spaced as the side that has fewer. So hopefully that makes sense. So diffusion is the movement of particles, while osmosis is the movement of water. Diffusion occurs when you have a lot of one side is very crowded, so you move molecules over to even out the amount of crowdedness on both sides. Osmosis is when you move fluids to make the space the molecules are in bigger, so they don't have to be so cramped together. Okay, diffusion is molecules, osmosis is water. Now back to your outline, diffusion. If the example we used earlier of diffusion, where the molecules were just able to move nicely from one side to the other, and then 1, 2, 3, 4, 5, 1, 2, 3, 4. Okay, so this example we used earlier is called simple diffusion. The molecules were able to just freely go from the high concentration to a low concentration. Okay, this was simple. It didn't require any energy. It was passive. Molecules were just able to float on until the sides were equal. This desire to move to the area of lower concentration is called your concentration gradient. Concentration gradient. That's just the desire of the molecules to move. So if you're in that crowded room with 50 people, that pull, that desire for you wanting to go to the other room would be your concentration gradient. The molecules will move if they get a chance, if their concentration gradient is pointing to the area of smaller concentration. Channel-mediated diffusion would occur if there was a channel involved in this. So let's add, here's another one. Let's say you have here a bunch of sodiums and a bunch of potassium. But you only have a channel that allows potassiums through. Okay, on the other side, let's say you have 2 of each. Okay, so both sodium, the reds, and the potassiums, the green, want to get through to the other side. But the only thing that's able to make it through this membrane is going to be that green potassium, because there's only a channel allowing potassium through. Okay, so the concentration gradient of sodium was still pointed in, it still wanted to come inside, but because there wasn't a channel that allowed it to come in, it had to remain outside. It was not able to enter into that cell. But the potassium moved freely until you had an even amount on both sides. Here's my channel. Okay, so the channel-mediated diffusion allowed the potassium to move, but not the sodium, because the channel was very specific on what it allowed to pass through. So this is going to be your channel-mediated diffusion. The top one up here was your simple. Okay, On this, it has examples: sodium, potassium, chlorine. I want you to memorize those because you will find them that you need to know them, okay? So memorize those. Facilitated diffusion is a lot like channel mediated diffusion, but it needs an enzyme to work. So this channel, is actually going to remain closed. Sorry, here we go. This channel remains closed until an enzyme comes by. So this channel is blocked off until an enzyme comes by, attaches, and opens up that channel. So the enzyme attached, the channel opened, and then that potassium was able to pass right through. Okay, that's facilitated diffusion. It just needed help to open that channel. It needed an enzyme to open that channel before it was able to pass. Okay, hopefully that makes sense. If you have questions, again, email me, whatever you need to do. Oops. Okay, on to osmosis. I'm going to draw 3 cups here. Super hard to draw on this. There you go. Okay, and then what happens? They're turning more interesting the more we go on. That's okay. Okay, so osmosis. Remember that was the movement of water that we talked about earlier? In our body, we have cells here. So each one of these is going to have a cell inside of it. Let's say this cup of water has lots of particles on the outside of the cell and very few on the inside. Let's say this one has the same on the inside as it does the outside. And then this one has lots and lots on the inside, but very few on the outside. Okay, different things are going to happen here. So we're talking about osmosis. So the particles themselves cannot move, but the water can. Okay, so the outside of this container is the one that's very crowded. It has a lot of people inside of it. It's this crowded room with 50 people. Since we're talking about osmosis, we're going to have water move to the place where there's more concentration. So water moves towards the place with more concentration. So since the inside of the cell is less concentrated than the outside, water is going to move out of your cell. This is going to cause that cell to shrivel up. Because as you get rid of that water, the cell then has less fluid on the inside. Okay, this is called a hypertonic solution. Hypertonic. That means that the outside of the cell was hyper or above the amount of particles that were inside of the cell. Isotonic solutions are ones that are equal. So what happens in your isotonic solutions is that the cell is actually going to remain the same since you had the same number of particles on the inside that you had on the outside. So you're still going to just have those 5 particles on the inside, 5 on the outside. That was a struggle, but you get the idea. No water moved because the same amount of space was found on the outside as the inside. Not one place had more particles than the other, because remember, water wants to move to the place with more particles. Hypotonic, I like to think of this one as hippotonic. The cell's going to turn into a hippo. So water wants to move to the place with more concentration, so water is going to move into this cell since it had more particles on the inside. Then that cell is going to swell up like a balloon. Until the particles have the same amount of room on the inside as they do the outside. This can actually happen so rapidly that the cell bursts. Okay, so fluid comes in and keeps coming in until the cell explodes. It happens sometimes. Okay, hypertonic, cell water moves out of the cell. Isotonic, no movement. Hypotonic, water moves into the cell. Okay. Active transport. Active transport is when you're moving something across the membrane. We're back to moving particles. So here's our membrane. We're moving something against its concentration gradient. So let's say we have 8 particles on this side and 3 on this. So naturally, you know that the particles want to move to the side with less, right? But this is active transport, so it's going to be a little different. You're going to actively move against your concentration gradient. This requires ATP. Okay, so this requires ATP. So ATP binds onto this channel. It's going to grab onto one of these particles and move it against its concentration gradient towards the side with more particles. So that's taking your room full of 50 people and the room full of 2. It's taking one of those 2 and putting them in the room full of 50 people. That takes energy. That particle does not naturally want to go into the more crowded area, but it's forced to using energy, or ATP. This is a form of channel-mediated diffusion, right? Because you have to have a channel to do this. You have to have a channel that ATP can bind to. So active transport is channel-mediated, okay? Our big example of this is going to be your sodium-potassium exchange pump. You're going to be pumping a bunch of potassium into your cell and a bunch of sodium out of your cell. So potassium always goes in, sodium always goes out. Okay, and that will become very important when we deal with muscles and action potentials. Okay, so that's active transport. It's moving against its gradient using ATP or energy. Okay, sorry, this chapter kind of jumps a little bit all over the place. But we're going to move to vesicular transport. So you have a membrane here. Let's say you have a protein coming up to this membrane, and you want the protein to exit the cell. What's going to happen is that protein's going to hit the cell membrane. The cell membrane is then going to form a little bubble for that protein to enter. So that protein pushes against the membrane, pushes a little bubble into the membrane until that bubble pinches off with the protein inside it. Okay, this is called exocytosis. Exocytosis. Wow, I can spell. There we go. Exocytosis. This requires energy. This takes energy to pinch off that vesicle. Okay? Exocytosis requires energy. The same thing happens coming from the other direction. So, whoop, whoop. So this is moving out of the cell. Same thing happens coming into the cell. So let's say you have a bacteria, bad guy, bacteria. It hits your cell wall. It's going to be engulfed by the cell until it's in its own little bubble on the inside of the cell. Okay, so moving into the cell is called endocytosis. So endocytosis is into, exocytosis is out of. Okay, exocytosis requires energy, endocytosis does not. Exocytosis requires energy, endocytosis does not. Endocytosis is also known as phagocytosis. Phago means to eat, so this literally means to eat something from the outside of the cell. It brings something from the outside and sucks it into itself. It eats it. It engulfs that cell or that bacteria, whatever is coming into the cell. Okay, mitosis, and then we are done. Mitosis, just know that mitosis is a word for cell division. I'm not going to go into detail about it because that's all you really need to know. Mitosis is just a word for cell division. So a cell that does not divide would never go through mitosis. Just use those interchangeably. Okay, stem cells are just cells that could become anything. They could divide into any different type of cell. They have not been determined what they're going to be. They could be a triangle cell or a square cell or a rectangle cell. They have not decided their identity yet. They have the potential to become any different type of cell. They're unspecialized. This process of them changing is called differentiation. They're differentiating into whatever the stem cell wanted to become. Okay, that is everything for that chapter. We actually kind of flew through. If you have questions, as always, just ask me, email me. I hope these are helping. I'm sure you guys all did great on your first exam. Good luck on bones and muscles. You guys are all awesome.